Welcome to the AgEmerge Podcast, brought to you by Ag Solutions Network. Your farming challenges are unique, so your practices should be too. We're here to share emerging ideas, build connections, and provoke conversation.
Get ready to improve your soil, your crops, your livestock, and your family's livelihood. I'm your producer, Kim Chase.
And I'm your host, Monte Bottens. Thanks for joining us.
Thanks for joining us. Today we welcome Dr. Jack Schultz, research professor in the Department of Biology and Biochemistry at the University of Houston. His research has focused on the chemical and molecular interactions between plants and insect herbivores. And those relationships are what Monte and Dr. Schultz explore as they discuss the amazing communications being discovered between plants and insects. It's a fascinating conversation, so let's jump right in.
Welcome, everyone. I'm pleased to be joined with Dr. Jack Schultz today. Welcome, Jack. I'm glad you could be with us.
I'm really happy to be here. Nice to meet you.
Well, there's everybody has specialties in communication and how they talk to each other. In fact, I had a minor in speech communications in college, but it was focused on human-to-human communications. Dr. Schultz, you've taken it a step further. You're listening to plants and eavesdropping on them. So tell us a little bit about your story, how you came to become interested in your field of study, and just what that has blossomed into your career.
I'm interested in biology of all kinds, and that's probably because I had parents that were outdoors people, hunters, fisher people, that sort of thing. We went fishing every year, and you can't be out there underwater every minute, so I had lots of opportunities to hunt for frogs and stuff like that. And my mother in particular was really interested in natural history, so I was aimed at biology from very young age. However, it wasn't until college where I, like everybody else who's a biologist, planned on medical school. I discovered that my quantitative and chemical skills I was not advancing in chemistry and math very well. And then I discovered ecology in my senior year. Some really outstanding ecology faculty took several of us on a trip to the Caribbean, where we floated around for a few weeks, counting birds and doing all sorts of insect things. And one of those professors convinced me not only that ecology was a really interesting subject with broad application as well as broad interest, but that insects were an ideal organism to work with for a number of reasons, one of which is they're little, you can culture them in many cases, you can do a lot of things with them. So I went off to graduate school intending to do something with insects, and I shouldn't say I discovered, we all know this, that one of the main things insects do is eat plants. So I wound up paying more attention to the plants than the insects. And when I finished my PhD and went for a postdoc, I participated in a variety of studies at the Hubbard Brook Experimental Forest in New Hampshire, where we were studying the interactions among birds, plants, or trees, and insects. We, for example, were the first people to show just how many insects the birds can eat in a period of time in a forest, and how much they depress the caterpillar population. But it turned out that birds aren't the whole story. Plants are defending on their own behalf. And we really couldn't explain which insects ate what plants and how much without paying attention to the plants, and that means plant chemistry. Now, you may recall that I said that I wasn't doing so well in college chemistry. As it turns out, this turned into a career focused on plant chemistry because that's what plants do to protect themselves against insects. So it was impossible to ignore. And a wrinkle that was thrown into it that really shaped our directions for the rest of my career was that if you just look at the chemistry of plants at the time that insects show up and start to eat them, you can get some interesting results. I mean, there are some chemicals in plants that are defensive right off the bat. But as it turns out, every plant that has been studied so far changes its chemistry in response to being eaten or attacked by an insect. And we call those induced defenses. And so, if we studied the chemistry of oak trees in a forest, that didn't actually tell us what chemicals were defending those trees against insects when they were attacked. So that opened, like a Pandora's box of messy chemistry and so forth. We devoted a lot of time to, and along the way, discovered that indeed oak trees change their chemistries in response to attack by insects, which at the time was a pretty big story because this was late 1970s, and the fact that plants actively responded to things like that was really new. So that turned into quite a good story. But it got even better. An undergraduate assistant and I attended a conference at which actually a friend of ours reported that he was finding that trees growing near to trees that are being attacked by insects also changed their chemistry as though they were being attacked, even though they weren't. Now, that was a very messy story because there were lots of alternative explanations that couldn't be ruled out. That was a field study. It's really hard to get solid answers that way. So when the undergraduate and I returned back to Dartmouth, where I was a postdoc, we did experiments in chambers with controlled air and controlled conditions and so forth, and found that indeed, if you wound, in our case, a poplar tree, it emits odors that can turn on the defenses in nearby trees. So that's where you got the talking plant story, right? Because that's the way it was described at the time. And it turns out that all plants emit bouquets, if you will, of volatile organic compounds, perfumes, if you want, that when they are attacked by insects. And what's really curious about it is that the specific makeup of that bouquet is specific to the insect doing the attack. So an oak tree attacked by one species of caterpillar emits different signals than an oak tree attacked by a different species. That started an entire new field studying the volatile organic compounds in plants, which has really, really taken off and has both applied and underlying basic significance for a lot of plants and a lot of systems. As it turns out, probably the more important function of those bouquets of odors is that they attract natural enemies of the insects doing the damage. So for example, corn plants emit one set of volatiles in response to one pest, but a different set in response to a different pest. And the significance of that is that each of those pests has a parasite that's looking for it, a parasitoid wasp, and it can use the specific odor trail to find that insect on that corn plant. If you think about it for a minute, if you were a tiny 1 millimeter person wandering around in a cornfield trying to find a stem borer, it's an impossible task. So what plants are actually doing with these volatiles is calling in their friends and making it possible for parasites and predators to control insect populations. And that happens both above and below ground, which ought to interest those of you interested in soils, because it's not as well studied, but it's very clear that chemicals emitted by plants below ground alter the soils in such a way that it can influence the resistance of the plant to various pests. Final part of that is that recent studies indicate that corn plants emit signals from their roots that attract entomophagous nematodes, that is, nematodes that kill root borers. And it's very effective over a space of about 2 meters. But unfortunately, domestication has bred that out of almost all corn varieties. Just recently, a laboratory in Switzerland engineered that signaling back into corn plants, and it increased their resistance to corn rootworm dramatically. So there are applications of these things, and that's just one of the examples. So that's where we got to. The bottom line is I'm really interested in how plants actively respond to things. The things I'm most interested in are pest insects. So it's plants, insects, and plant behavior for me.
Fascinating, fascinating work. In fact, I think we should provide free aspirin for anyone that listens to this podcast about how mind blowing your research is. So,
Well, you know about aspirin's original source.
true. Good point.
It's willows, right?
Hmm.
Uh, uh, Aspirin is just a slightly modified version of a chemical that's defensive in willows. Uh, Native Americans used to chew willow bark to alleviate pain. And Dr. Bayer actually found a way to make it more effective in humans, and that's why we have aspirin.
Right.
Tell me, do you drink coffee?
Of course.
And people who drink coffee are interested in the alertness effects, and that's because caffeine acts in the central nervous system. But if you think for a minute, plants don't have central nervous systems. So why in the world are coffee plants making caffeine? And the answer is it's an insecticide. It's both lethal to small insects and it's very, very, very bitter. So it does a pretty good job protecting coffee plants and the beans from pests. The better studied example is nicotine, which has been used as a pesticide for centuries. Uh, and that's a really good example of the induced defense situation. When a tobacco plant leaf is wounded or attacked by an insect, signals are transmitted to the roots where nicotine is manufactured. The nicotine is then pumped up to the site of damage and increases the resistance of the plant to the attacker. That's economically significant because at least at one point, at least some growers used to top their tobacco plants because that increases the nicotine content of those leaves. So there are all kinds of connections between this basic science, which I follow in part largely because I'm curious, and applications.
Well, I love the curiosity because, that's an interesting mechanism on the tobacco plant because it's produced, in the root area. Yeah. And there's significant microbial interactions that happen,
Oh
for
yeah,
that. So, very fascinating. So you didn't like organic chem and look at what you did.
yeah. Well, I had lots of good collaborators.
You found people who liked organic chem. There you go.
And we're good at it.
All right. So let me take it one step further. You mentioned did you look into outside of insects? Is that when you're mentioning oak trees and infested by caterpillars and such, is there signaling happening to mammals, birds?
Yes, actually, just in the last half dozen years, investigators in Finland demonstrated that birds are helped in locating caterpillars on, I think it was willow trees, by following the odors, which is really startling because for decades people assumed that birds had, very little sense of smell. It turns out that finding produced a line of research that found that actually birds have a pretty good sense of smell and they can definitely use those signals.
Hmm. All right.
I
Take the aspirin
recently,
and eat the willow bark.
yeah, well, yeah, I recently visited Australia and took a tour of rainforest there. And this kind of thing happens to you when you're a scientist and you do these tours. The tour guide, when he was introducing us to the forest, said, uh, actually, the way this forest works is that all of these trees here are communicating with each other through, airborne signals. Um, and so everything here is connected, which is people like to say. Um, but so I'm standing there thinking, oh, I know about that, you know. Uh, but the interesting part of that was that he claimed that the native people there could tell what was going on in the forest based on their perception of those odors. So if there was, a tree that's really being damaged heavily or something, they could tell where it was. And I thought that was fascinating. They're really, as far as I know, there are no studies of this sort of ecological signaling by plants to humans in that way.
There's something interesting about that because, you can, and maybe you've observed this, but you can walk into certain fields and you can just sense something's wrong. I don't know
Oh
what
yeah.
it is. It's
Mhm.
some combination of our senses is, and you can just you get a feeling and kind of go for it. And I think a lot of times we just have to pay attention to our senses and not dismiss them, right? Sometimes
Oh,
we
absolutely.
sometimes we think too much for our own good.
The AgEmerge Podcast is brought to you by Ag Solutions Network. The ASN team is hands-on, digging in and invested in regenerative agriculture. Along with the proper plant nutrition and biologicals to boost your soil microbiome, we provide the ideas and implementation guidance to support you on your soil health journey. So stop farming the same way and contact Ag Solutions Network today at asn.farm.
Yeah, senior year, a trip to the Caribbean, that will certainly have an influence on your career.
Oh
That
yeah.
was a pretty good thing. And thankfully you were this close and you avoid the entomologist thing. So, that means you still have a good personality, right? And I
Hey, I have a hat that says retired entomologist. I mean, there's no escaping it. I was a
oh,
professor of entomology at Penn State. So, but interestingly, the entomology department at Penn State is heavily invested in chemical ecology. And so many of the faculty there are in the same boat I was.
yeah. Yeah. So let me ask you, one of the things that's really plaguing that area is ash borer.
Yeah.
And doesn't seem to be able to be stopped. We're looking at, again, something else to eliminate a species. We have a signal interruption there, don't we? How do we help it signal or something to stop the progression of that?
Well, the first thing is you need something that's interested in those beetles. And generally, when pests are carried from one continent to the other, their enemies don't follow.
Hmm.
Of course, the USDA has a huge effort looking for natural enemies of a wide variety of pests. Um, that effort, although a good idea, is rarely, well, it can be successful, but it's very, very difficult. I think something like 300 natural enemies have been introduced to control the gypsy moth on oak trees. Um, and what finally worked there was a fungus. So, and it took a long time, and that fungus occurred naturally. Anyway, that beetle probably didn't bring any natural enemies with it, so there's nobody to follow the odor trail. First of all, second of all, although boring insects, and I don't mean to say it's uninteresting, I mean it bores in the tree. um, boring insects can cause plants to emit odors, but it's a little harder because they're boring in wood, and living tissue connections are not so good with the leaves, which have to emit the odors. Um, but there are people working on resistance traits, trying to find tree genotypes that are naturally resistant. I mean, the standard thing that we do there is we try to identify the bases of resistance, chemical first and then genetic later, by looking for resistant trees. I mean, it's the same way you do in breeding, is to find resistant cultivars and so forth, except that this is mostly naturally done. But then what do? Well, agriculture has tended, for a variety of reasons, to focus on and use resistance as they find it. The problem is that at least Western agriculture then plants enormous monocultures of these resistant plants, and I can tell you that there is no such thing as forever resistance. Insects and microbes are so fast to evolve the ability to deal with that stuff that you really can't call anything ultimate resistance. And you just make it worse by feeding them an enormous field or enormous, acres of plants that are identical in that way. Yeah. Um, so the alternative to that, there are actually two issues here. That's one. Uh, the other issue is that plants have budgets just like you and I do. Their budgets have three main concerns: growth, reproduction, and defense. And that's a finite budget. Unless a plant can increase its income in terms of photosynthesis and carbon, if it invests more in one of those functions, it has less to invest in the others. So by maximizing reproduction, that is productivity or growth for crop purposes, the defenses naturally take a hit because the energy's got to, and materials have to come from somewhere, and that's where they come from.
Mhm.
So most of our agricultural plants have had these defenses lost, either because of economics, of the plant's economics, uh, or because of selective breeding. You've probably seen pictures of the original tomato, right?
Yeah.
It's about the size of a dime, and it's all hairy, and you really would not want to eat it. Uh, Today's tomato is quite different, but that came at the expense of the defensive chemistry of tomatoes. Now that's a good thing for eating tomatoes. You really don't want to eat the defenses of tomatoes as they originally were. So for both taste and health reasons and productivity, we've lost most of those defenses. You come back to something like ash trees, and we don't really know anything about the basis of their resistance to things. This is a brand new insect, so does it even have defenses that are relevant to this thing? If so, what are they? Are there underlying genes that you can deal with? That's why so many researchers are really interested in gene editing as the future here, because um, with gene editing, and I don't mean necessarily transgenics, I mean the ability to go into the genome and change the expression of individual genes, turn them up, turn them down, change their function a little bit. That has less impact on the economy of the plant. And it's not really the kind of thing that people are afraid of. You're not actually putting like, I don't know, a firefly gene into a corn plant. You're actually just tweaking the genes of the corn plant as they naturally function. And a lot of researchers are very enthusiastic about that. In fact, there's been some very successful research doing that to increase the photosynthetic capacity of plants, including crop plants, which would increase that budget and give the plant more to work with, so that it might be able to, continue to be productive and still defend itself. But boy, it's really there is a researcher at the Wayne State University doing a lot on trying to find resistance to the borer, but it's very slow. Trees are really hard to work with. They don't reproduce until they're like 50 years old. Some of them. That doesn't make for a good lab rat. You're not doing much crossbreeding with those.
Well,
Oh,
let's talk a little more about lab rats here. So, you've got, and one of the things that I think about is, we do a lot of things in agriculture to get a benefit, whether it's yield or plant defense mechanism,
sure.
those, herbicide resistance and such. But
Mhm.
then there becomes the unintended consequences. So,
Oh,
got
yeah.
all of these corn plants out there. They're all talking to each other, and then we insert some transgenic traits, which is what's being done today.
Sure.
Um, how does that affect the signaling between plants? Does it effectively, maybe because we're changing a lot of the metabolic pathways when we do that, we're going to be changing organic compounds that they produce? Are we hitting the mute button on those plants to be able to call in the predators to defend them, or signal that, hey, I've got a problem, get ready?
Yeah. I've got two answers to that. The first one is it kind of depends on what you tinker with.
Hmm.
Um, so let's talk about cotton for a moment. Cotton's defenses include fairly large concentrations of condensed tannins. Those are protein inhibitors. They make it difficult to digest the plant, and they're actually bad tasting as well. Um,
But
if you tinker with,
just like the coffee, in wine, it's good, for people. It's
oh sure, oh yeah, absolutely.
deadly to insects, good for people. I'm seeing a trend
N-
here.
no red wine is truly great without tannins.
That's true.
That's no question about it. And the tannins are protective in grapevines too.
Mhm.
Um, but when the fruit ripens, the tannins drop, although less so in a red grape than in a white grape. That's so birds will take them.
Mhm.
Anyway, but tannins, that whole biosynthetic pathway, the chemicals involved there, uh, are rarely or never volatile and generally don't participate in external signaling of any kind. So you can tinker with those and not change the communication much. On the other hand, the plant also produces terpenes. You want to think the odor of pine sol or pine trees. Those are signaling compounds. They turn on various things in the plant and in other plants, and they're attractive to natural enemies. They're also attractive to bark beetles, but that's another story. If you tinker with those, you run the risk of modifying the signaling system. So that's a little trickier, a little more dangerous. So that's answer number one. Answer number two is, as you kind of alluded to, and I'm kind of at odds with the bioengineering community on this. The more you study the gene networks in a plant that govern the various functions a plant has, the more connections you find. Now, I know it's a kind of a corny ecologist saying, but when it comes to gene networks in plants, I really think you can't just do one thing. Right now, I'm studying plant responses to insects that form galls on them. I don't know if you know about galls. You know about Hessian fly in wheat?
Yes. Yes.
All right, the larva there causes a swelling on the plant. That's called a gall. That gall is a strong sink. It draws resources from the plant, particularly the upper part of the plant, and that's why they lodge. They fall over because the stems are weak. That's a change in the development of the plant. Those insects that do that, and I work on phylloxera, on grape leaves, those insects actually change the expression of genes involved in the development of the plant to produce novel structures. Some of them are absolutely wild and crazy, with spikes and red colors and all kinds of crazy things. So the insect's tinkering with developmental genes. But the more I look at those developmental genes, they're also involved in 6 other functions. So as I think you were thinking, strikes me as a little arrogant to think that you can just change one gene or a couple of genes in a plant and alter only one function. I've got my doubts about that. And now that's different from inserting whole new genes like Bacillus thuringiensis toxin in plants. In cotton, that's an interesting story. The condensed tannin, we showed that condensed tannins block Bt function in insects. They bind to the protein and prevent it from having its effect. And the ag chemical company came to talk to me about that. I won't name them. And I was a young assistant professor, and I just blurted everything I knew. And the next thing I knew, what they had done is tripled or quadrupled the number of Bt genes they were inserting in the cotton plants to overcome the condensed tannins. So the expression of Bt genes in cotton now is really, really high because cotton naturally resists their function. So if you're going to tinker with these things, you got to know what you're doing. You got to know lots about all the pathways. It's really complicated, and it keeps some of us in business for a career.
Well, see, there's an unintended benefit, right? So
yeah. Well, ecologists have known about unintended consequences for decades. That's what they're about. You do this thing over here, and you remove the wolves from Yellowstone, and something happens to the trees. I mean, that's the kind of thing they're used to seeing. Right. The same thing is true in lots of other settings.
Yeah, that's very true. So, one of the things that we notice on growers we work with that plant non-GMO corn, for example, less incidence of tar spot. Okay. Why is that related? Totally different things, less incidence of insect issues. So it's fascinating. Let's one other thing to talk about on the unintended consequences, where we're maybe disrupting the calling, is I think a lot of times with our modern seed treatments where we have insecticide load in a lot of the plants in a field. If a plant does get attacked by an insect, we're also taking out some of the other beneficials that would be there to respond. Is that almost the equivalent of calling but nobody's home? Where that mechanism is.
Well, sure.
We just don't
if you knock out the
robustness.
Sure, if you knock out the beneficials, there's nobody to respond. It's actually the interaction between induced defenses in crop plants and the application of pesticides is a pretty active area now. I was just looking at a paper on that this morning, sort of investigating both the chemical and ecological outcomes of mixing those two things. And guess what? It's complicated. It's not, and that's really a very young interest in people. And so there's really nothing to be said yet about what the actual interactions there would be, except you have definitely identified the fact that if you knock out the beneficials, there's nobody to respond to the calls.
Right.
People are interested in whether the chemistry of pesticides actually interferes with the chemistry of signaling too. But that's just starting that kind of research.
More and more cans of worms to open here.
Oh yeah. Yeah, mine was a
Literally.
40. I've been doing this for 40, 45 years, and I'm still analyzing data.
But no, it's interesting to think about all these little things that we do. So now on the beneficial side, we've been incorporating cover crops a lot, and especially in orchard crops. And we are noticing significantly reduced needs for miticides, insecticide sprays, even
Yeah.
fungicides. And now I can understand the insecticides because we're providing a habitat to bring beneficials in. So when the tree would respond or send out the signal, hey, we've, instead of waiting for the population to increase and respond, it's there to respond. So it's a speed thing. But we're also seeing some things in regards to fungicide and bloom and those kind of things. Would that be more of a root signaling response? Would you just purely anecdotal here, but we are
Well,
seeing less fungal issues on bloom.
there are some less anecdotal stories. And by the way, when I tell you that somebody knows something, I'm really talking about published stuff that is backed up by reviews and stuff. I really,
Right.
having done public communication and science for a long time, I'm really irritated by just so stories. Anyway, but here are two, well, they're related, that apply to your interests there. Japanese investigators showed that if you grow bean plants in, pots and soil, you add aphids to them so that the plant responds defensively to the aphids, and then you remove that plant and plant a new undamaged pea plant in the same pot, it expresses the defense responses. So there was definitely signaling left behind in the soil that the undamaged new plant sensed and caused it to turn on its defenses. And that is also true of volatile emissions. You get a pea plant in a pot to emit its volatiles, you remove it, you put a new plant in the pot, it will emit the volatiles. So there's definitely below ground signaling going on there. I already told you about attracting nematodes. As I'm sure you know, it's really hard to do chemistry in soils. They stick to everything in the soil. It's really a very difficult system. But more and more people are finding that characteristics of the soil are critical to how the plant can respond to things.
Think about, I was thinking that when you were telling your story in the 1970s, when you did your first work there on the oak trees, look at the technological advances we've had in sensors and lab ability to do this. And now it's just exploding, isn't it? I mean,
it is. But we've been limited. Well, first of all, let's back up for a second. Everything we're talking about here happens as a result of changes in the expression of genes. In order to make these signals, plants have to turn on genes that make the proteins that make the signals. So I've gone from a situation where my major piece of gear was a butterfly net to doing almost exclusively molecular work now. And the reason for that is I wanted to know how this works, and the answer is in the genes. Well, now we can do that. So the question for the pea pot experiment is going to be, which genes are turned on or turned off by whatever's in the soil? And we can now do that. However, we can still only do that mainly with a few model plants because we don't know the genes in most of the plants. The whole reason I got into the molecular world was that I wanted to know how the oak trees were responding to these things. and there was no oak genome. In fact, I don't think there still is an oak genome. So there was no library of genes I could go to see what was, what was active and what was not active. That was very frustrating, and reviewers at the National Science Foundation made it very clear to me that I was not going to get away with that. Boy, those some brutal reviews. What are you talking about? You can't do this. Nowadays, you can sequence an entire plant. In other words, identify its entire genome for a couple of $1,000s in a week. Now, the problem with that is we still don't know necessarily what each of those genes does in that plant. And so we're still limited by studies that are needed to identify the actual functions of those genes, even though we can find them. And that's still best done in a couple of model plants. That's why I work on grape. I've worked with Arabidopsis, which is the lab rat. And there are a few. Agricultural plants are coming on. USDA has slowly rotated its preferences and funding away from model plants towards more crop plants. And at the start, that was very frustrating because you didn't have anything to work with in crop plants. Nowadays, though, the genomes of a lot of crop plants are sequenced, and we have a lot more to work with. Some of them, like wheat, though, are really still difficult. I think wheat is a hexaploid or something. I mean, it's got a nutty genome. But those technical advances have really allowed us to do a lot of things we couldn't do before. And you're absolutely right. We wouldn't know half of what we know now without them.
So let's dive into that a little bit. Do you think in the future, will it be easier to identify these compounds, these defense compounds that they are making, or will it be easier to identify with some sort of remote sensing technology genetic markers that have been activated?
Mm-hmm. I had a couple of grants from DARPA, the Defense Advanced Research Agency.
Mm-hmm.
Okay. And what they were interested in was whether or not plants could report their exposure to TNT. They were wondering whether soldiers could measure something in a field before they walk through it to see if there were landmines present in the soil. It turns out that plants take up TNT really well. In fact, it's kind of it's sort of protein-like, and they can use it to improve their growth. So the idea was, and TNT leaks out of landmines a lot into the soil. So the idea was, if there was something you could identify that you could measure in plants, they might tell you that this field was mined, or even where the mines are. Well, that's a gene question as well. Okay. Our approach to it was to see whether or not plants emitted a characteristic odor bouquet in response to TNT in the soil, and they do. You could definitely locate a landmine if you could sample the odors, the volatiles emitted by the plant. The military is also interested in whether plants could help us find biowarfare agents. We didn't have access to there are only 2 labs in the country where you can work with serious bioweapons. And we didn't have one, of course, at Penn State. And so we worked with the closest relative to anthrax. It's just a few genes different from anthrax. And we found that plants exposed to that bacterium also emitted a characteristic odor plume. So in both of these cases, you could detect the presence of that biological agent and or TNT by sniffing the plants. The problem with that is the only thing on the planet that is that good at sniffing plants is a dog. And dogs can already smell TNT. I mean, they can be used in demining, except that nobody wants to lose a dog. What's happening now in I think it's in Africa is the use of rats, which also are really good at sniffing TNT, and they're very helpful in finding mines. And I guess, and forgive me for saying this, but I guess people don't mind losing rats here and there. I don't know. But nobody wants their golden retriever blown up, So dogs have something like 400 times more odor receptors than we do. Their world is, they see the world through their nose. And so it's no surprise that they can identify a plant that's infested with something or growing on TNT, no problem. So we figured, well, what can we do with that? And the only instrumentation that humans use for that is a gas chromatograph, which is, a kind of instrument that pulls the air with the stuff in it in the front end and separates all the components and tells you, what was there. The problem with them is that you couldn't just walk through a field, like a soybean field or something, and expect it to be able to register at the concentrations that these things are out there. So we worked with an engineer who's now at the University of Michigan, who developed a gas chromatograph about this big that we thought might work in the field. I mean, you'd have to, well, what we thought is you might put it on a tractor or carry it around in the field, and you would arrange it to light up when it detected something you were looking for. And then we ran out of money, and we got to the point where we were going to, well, first greenhouse test and then lab test that, and the DARPA money ran out, and NSF wasn't interested, and so we never got any farther with it. That machine now is doing another thing that dogs do in diagnosing human diseases by sniffing the breath of patients. You can identify diseases that a person has from the odors that they emit. And dogs can detect that, and now this gas chromatograph is used for that. So we got something useful out of it,
Okay, so let me ask you that defense mechanism is that the human that is creating those odors, the organic compounds, or is that the disease creating those organic compounds? And to what end was that done if there's nothing that can react to that organic compound, I guess?
Well, first of all, it's a plant making these things, and it's doing that in response to something. Mm-hmm. Why exactly it emits something that you can identify in response to TNT in the soil, nobody knows. Mm-hmm. In the case of bacteria, plants are exquisitely sensitive to bacteria and can identify them by single gene differences. They're really good at that. The question is, why would they emit particular volatiles in that case? And as it turns out, one of the very first lab studies of signaling between plants showed that you can create virus resistance in uninfected plants by exposing them to the air from infected plants.
Say that again.
You can
Very important.
You can generate resistance to viruses in an uninfected plant by exposing it to the air from an infected plant. Now, that gets us back to the talking plants and are they trying to help each other? It's, an explanation for that, might be that viruses spread well, and maybe the emitting plant, cuts down on the spread of the virus to other plants, but it's really not in an individual plant's interest to help its neighbors. They're competitors.
Correct.
So it's not immediately clear in that particular case what the signaling means. We do have an alternative explanation. In another study we did, we found that when a leaf, say, if you count the leaves from the top of the plant to the bottom, 1, 2, 3, 4, 5. If leaf 5 is attacked by an insect, the airborne signals from that leaf 5 get to leaf 1 much faster than any signal that can go through the plant. So it may be that the function in both the virus case and the insect case is that the plant is helping itself by spreading its own signals. It's talking to itself, to speed up its responses. That's a possibility.
I think our human nature still loves the warm and fuzzy feeling that it's trying to, the plant's trying to protect its neighbors and the survival of the species, right?
So I know there's been some argument about that, but yeah, I mean, the way natural selection works.
Then you get the sociologists involved, and then it all gets off the rails. So.
Do you know how often I have to have this kind of conversation with people who believe in conscious plants?
Yeah, yeah. Well,
Yeah, yeah.
you yourself, I do have you on record saying, quote, plants are just slow animals. So,
Yeah.
I
All right. So
Other-
that's a bit of it's a bit of an exaggeration, but I'm telling you, I'm on a crusade to get people to understand that plants have behaviors and they're
yeah,
much more dynamic than you think. Uh, there's a thing called plant blindness, and I think people are objecting to the use of the word blindness. I don't know. But when I give a public lecture, I always ask my audience, did they notice the plants in the lobby on the way in? And nobody ever does. Of course, sometimes there aren't any,
yeah.
uh, but if there are plants, nobody noticed them. And that's the way people think about the plants. They're part of the furniture. And is that a real plant to your left there?
No, it is not. See,
Okay,
I
so
it's we're in a
it is furniture.
non-lighted environment. It is furniture.
Yeah,
Yes, you're looking at furniture all around. So,
yeah, okay. Well, yeah, every chance I get, I try to convince people that plants are more dynamic and do more things than they thought. And that animal comparison is kind of an exaggeration. Although, if we went through, step by step, particularly in terms of senses, the comparison would be pretty good.
hmm. So a couple other things you mentioned. Your earlier the machine that you were saying was about 6 inches in size.
Yeah,
You were also involved with something else that got these individual compounds down to sensing something about the size of a chip that's the size of your
right,
finger or so
right.
to identify specific compounds. And then talk to us about that, what that is, and how that can translate into, particularly like fruit ripening and such, because we have a lot of
Mhm,
tomato growers and vine growers
mhm.
that listen to the podcast. So share about that technology and has that progressed any to a
No,
commercial scale?
no. First, let's talk about that, and then I want to talk about what you could do if they do work. And to be honest with you, I think the possibility is there, but I'm not an engineer. It there are a
You
lot
only
of
do so much.
yeah, well, there are a lot of sociological things that influence how science is done. And one of them, in my experience, is that it's not that easy to get engineers interested in a fundamental biological question.
Hmm.
And then if you get them interested, it takes some work to get them to understand it. So I was lucky to find one or two, who would take a shot at this kind of thing. But the other sociological thing that comes in is you run out of funding or accidents happen. So the first thing we tried with that is a metal chip uh, that vibrates at a particular frequency when you expose it to an electrical force, so a radio wave, actually. Okay. If you coat that chip with things that capture things flying around in the air, the increased mass on the chip will change the frequency at which it vibrates when you hit it with a radio beam. Okay. That's used in lots of settings. So I got an engineer interested in whether we could design one that would change its frequency when it was exposed to plant volatiles. It turns out that the polymers that you coat these chips with are difficult to make, and they have to be designed so that they have a specific affinity for the molecules you're trying to find in the air. So he assigned a postdoc to that project, and creating these polymers is done in a safety hood because they're toxic, as you're making them. And they had a hood failure, and the postdoc was poisoned and wound up in the hospital for months. In the meantime, and here's where the engineering culture comes in. In the meantime, uh, the engineer discovered that the chips you can feed them to people, and as they pass through the gut in a capsule, of course, when they pass through the gut, they can measure the pH of the gut and tell you all about the pH of the gut from the top to the bottom by passing through, as long as you hit them with these radio waves. And that was a much more profitable application.
Hmm.
At the time, there was no money in creating a chip that could sense plant volatiles, but lots in medical applications. So we lost that one. I already told you about the other one that ran out of money. So the various devices that I've described have been partially developed but never finished. So it's frustrating, and you need a particular team. It's absolutely an interdisciplinary problem, and you need the right team to do it. I haven't been able to put it together. What could you do? Well, it's already been distinguished. When you go to the market and you're looking for a ripe melon, how do you do that?
Well, you look
You
at
can
it.
thump it.
Yeah, you can thump it. You can look at it. You can smell it.
Uh-huh. It turns out that a gas chromatograph is much more accurate at telling how ripe that melon is than your nose is. This has been demonstrated with grapes, where it's critical to know exactly how ripe the grapes are when they're harvested. And it turns out that experimentation shows that if you can analyze the odors from the grapes, you can detect their ripeness much more accurately than you can looking at them or using your own nose. So there's an obvious application. Grape growers are really excited about the possibility of better ripeness, measurements, because you go through the vineyard, and they're all different stages of ripeness. A big problem they have is figuring out when to harvest and which ones to harvest. The same thing is true in a greenhouse. If you look in a tomato hothouse, there are green tomatoes. There are more ripe tomatoes, and there are ripe tomatoes, and then there are the diseased ones and the ones on the floor. They all smell different. If you had a small enough device to sniff that, you could identify the ripeness of each of those really precisely and make a decision about when to grab them. So sorry about that.
No, that's interesting, and how I'm just thinking that you made the comment of two examples there that just got lost in the shuffle, either from a better application, a higher value, or lost funding, or those kind of things. I'm just imagining how many unfinished projects are out there with good technology that are not being applied, that just gets lost.
Well, okay. My career has been pretty much driven by curiosity. So I'm a basic researcher. And I really promote the need for basic research, which has many fewer funding sources than applied research does. But it also has no constraints on what you can find. I know from your previous podcast that you're familiar with the problem that applied researchers run into when they're funded by industry. Industry is interested in a particular outcome. In academia, if you can't publish your work because industry prohibits it, you're not going to get tenure. So that's from my point of view, that's a very dangerous situation. So I fully believe that we need the kind of information that basic research provides to carry it forward into application. Without that, basic research is turning up the bottom line, functions and principles that you can then take into the field or into the greenhouse to work out its uses. A lot of people get irritated when you say, well, I'm interested in this because it's interesting. Well, that's true. It's also interesting because it has an application. But all my funding has been from the NSF, DARPA, and some grants from USDA and the Forest Service. And I've always highlighted what potential applications could come out of our work. But we've been working pretty much at the basic level.
Well, it has to be done, right? We have to satisfy that curiosity, and I appreciate your curiosity. And while we still got a little bit of time left here together, I wanted to share with you an interesting observation we had on our own farm. About, I forget, 10 years ago or so, we had a real bad outbreak of Japanese beetle on corn, in Illinois, and it's continued to be a problem ever since. And I happened to be in California at the time, and I was getting these reports from University of Illinois Extension. Oh, check, it's the world's coming to an end. And we had been out and sprayed one of our foliar products that contained a lot of there was kelp extracts in it and some other signaling compounds. And my dad was saying, I don't see any. There's maybe one or two. I just don't see any Japanese beetles. And then he would walk across the fence into the neighbors, and only like 4 or 5 rows away, the plants would be covered with them. And that was when I first I started digging into it a little bit and realizing that, hey, there's signaling happening,
Mhm.
between these plants. And then we would see a hail event on one field, the same variety across the road didn't get hailed, but then the Japanese beetles were in where the plant had been stressed by hail.
Mhm.
But the unstressed plants didn't have it. So
Mhm.
eh, there's just this amazing potential for application, isn't there? I mean,
Oh yeah,
why
everything that
kill the
happens.
bug if you can just scare it away or make the plant
Well,
invisible,
they will
essentially?
go to your neighbor.
Don't tell him.
Well, two things about that. One is we've talked mainly about these signals attracting beneficials, but they also attract pests.
Yes.
You know, sometimes it's the same signal, but often it's not. There's one for the pest and one for the enemy. Uh, and Japanese beetles are very sensitive to that. They also aggregate using their own pheromones, and that's why people use pheromone traps for them so much in gardens. If you put them, but what a lot of people do is they put the trap in the garden,
Oh,
and the
and attract
beetles,
them.
yeah, the beetles, they quickly fill up the trap, and then they're on the garden.
Uh,
Um,
so put the
so
trap on
yes,
your neighbor's garden. Okay, I got it. Yeah.
that's right. Yeah, well, or some distance away to be kind. Yeah, that's true. But let me tell you about a very successful approach using the observations you just made. There's a technique called push-pull technology that's been very successful in Africa. I don't think we'll have the time to go through it here, but it employs mixed cultures in which some of the plants repel pests. Others are planted in such a way that they can attract pests away from the crop. And yet others are there because we know that they attract beneficials. So it's a mixture of plants using what we know about their specific signaling systems to protect the crop. Evidently, it's very successful, but the catch is that's not modern agriculture. Those are, family farm, small plots where they can do mixed cultures because they don't do any automated harvesting. And automated harvesting is a real enemy of doing any of these things because if you have to have uniformity, it really makes it hard to get some of this stuff done. Although plants can be physically uniform and chemically very different, so
Yeah.
that's the place to go, I think.
But back to talking about those, obnoxious engineers. They're working on things now with automation,
Yeah.
sensing technology, and that'll make it smaller. That
Hmm.
that we can then have companion crop growing situations that we can mechanically harvest.
Yeah, I noticed that the
There's some
harvesters
neat
are
things.
getting much more sophisticated. Yeah, and I
The
don't know.
one.
Have you seen the research at Davis where they're using video and lasers to kill weeds early in the crop?
Yes, yes,
Mhm.
and that's a great way to do it. We've had one of the folks on the podcast about that,
Oh yeah.
and I think as we get that polyculture involved, there's soil benefits as far as water availability. There's definitely plant health benefits, and really it's a return to what the Native Americans did, right? Except for we're able
Oh
to
yeah.
mechanically
Right.
harvest instead of hand harvest, and um, one of the
I've,
things
I've,
about the harvesters
I've
just get
Go
bigger
ahead.
because there's fewer people to run them, right?
Well,
So
that's true too. Yeah. 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 I've spoken at a couple of soils and cover crop conferences, and something nobody's paying attention to is the kind of observation that we talked about earlier, in which the characteristics of the soil influence what the above ground plants do, including defenses and interactions with enemies. If we knew enough about what particular plants we're putting into the soil to turn on, defenses in the crop plant, or what they're doing, to kill pests while they're there. And I think that's done a little bit because you can figure that out practically. You could really select uh, cover crops much more intelligently.
yeah, select.
I realize a lot goes into that, but I think one thing that doesn't get as much attention as it could is what the cover crop does to the soil quality in terms of defenses.
So almost like the genome for the plants, we almost need the effects of the plants as
Yeah.
far as are they calling in beneficials, are they repelling pests, and
Hmm.
create that cocktail to where that is the cover crop and or the companion crop that we're running
Hmm.
with our
Hmm.
cash crops to make that happen.
Now, let me warn you,
Yes.
tobacco has tobacco hornworms, and so do your tomatoes, right?
Mhm.
They don't care about defenses. Uh, and they're really good examples about how for every measure, there's a countermeasure on the part of the pest,
Right.
Uh, Tobacco hornworms even emit gaseous nicotine from their spiracles to scare away spiders.
And that's their primary predator.
That's one of them. Now, birds
Huh.
would be the primary one, but birds aren't going to touch those things. They're really awful.
Well, they're loaded with nicotine.
That's right. Yeah. Yep.
Well, nature has a way to overcome, like you said. Nothing is resistant forever, and everything is a food source for something else. So, anyway, no, I really appreciate the work that you've done, and I love the curiosity part. You mentioned it multiple times, and that being curious and just asking why is so important. And I just really appreciate what you've done and how you've contributed to the body of science. And we can hope that it'll more and more of what you've done will get picked up and put in applications. So,
Yeah, we hope so too.
anything else we should have added while we were visiting here together today?
I don't think so. I think we've had fun.
We've whet the appetite, and we'll certainly have some resources, and Kim will put in there to follow up and look at the vast amount of work that you've done, and people can explore and dive deeper on their own. So I really appreciate it.
Well, it's been a great time with you. I really appreciate being here. Thanks so much.
Thank you, Dr. Schultz. Take care.
Thanks for listening today. I am amazed at the way plants and insects interact, and I think it's so critical for us to understand these relationships so we can utilize them in our soil health systems. And as always, we'd love to help you implement a systems approach to healthy soil practices. So check out our website at asn.farm, and there you can click on links to follow us on Facebook, Twitter, Instagram, LinkedIn, and YouTube. There's a lot of great things happening and always something to learn. Thanks for listening.